Good morning, everyone. It's my pleasure to introduce one of the department's research faculty for Grand Rounds this morning, Sara HamiltonHart. I've known Sara for an awfully long time. I shouldn't really need to get a copy of her CV to introduce her, but I'm really glad I did ask her for one. So it's really impressive looking at all you've done since you've been a junior faculty, Sara. So, for those who don't know Sara yet, she got her PhD at the University of Iowa with John Hardy, and then came up here and did a postdoctoral fellowship with our own Steve Jameson. And then she started her own lab in 2014, and she was appointed in the department as an assistant professor on the academic track with a research focus. Her lab is over in WMPB, focuses on the immune response to malaria, as we'll hear about today, but also other infectious diseases. Sara has a very impressive publication record. Her work's funded by 2 NIH R01 grants and an NIH supplement. She teaches and is a graduate faculty member in the MyCab graduate program. She served on numerous NIH and a VA grant review panels. She is the director of the University of Minnesota's Dirty Mouse Colony, this facility that tries to mimic normal microbial experience in animal models, which she may or may not be talking about today. I don't know. She is also currently serving on the Institutional Biosafety. So, Sara, pleasure to have you this morning.
Thank you, Chris, and thanks everyone for being here this morning. Last time I gave Grand Rounds, I talked about dirty mice, so this time I'm not going to talk about dirty mice. I thought I would talk about some of our other work in the lab where we're investigating the immune response during severe malaria. And I'm sure, as all of you are aware, malaria really continues to be a very serious global health problem. And in 2017, there were 218 million cases of malaria and 500,000 deaths resulting from that infection. Interestingly, we used to have malaria in the United States, but with the use of DDT, it was eradicated from the southern United States. So today, it's predominantly in areas of Africa and also Southeast Asia and South America. The immune response and immunity to malaria develops over many years and multiple exposures. That's one interesting thing about this disease, is that it takes a long time to develop immunity. And so the most vulnerable people are young people that have had very little exposure, and that's where you get really high levels of parasitemia and severe complications. And then over time, you'll see that the clinical symptoms diminish as people age and you get that repeated exposure. But interestingly, we never achieve sterilizing immunity. So people, even several decades into life, will carry parasite load, but they will not experience the symptoms of disease. And so the immune response is most simply kind of divided into 2 components. Antibodies directed against parasite antigens at various stages of the disease, and also CD8 T cells, which through their cytotoxic functions and interferon gamma production, direct that against parasite-infected cells and reduce that parasite load. And there is one vaccine that continues to be tweaked and tested in clinical trials. It's not a great vaccine. It's called RTSS. It's directed against the protein on the parasite, And a problem with it is that it takes 4 injections to reduce disease severity by 25 to 36%. So there's definitely a lot more work that needs to be done to improve this statistic. And it's also been found that efficacy wanes over time, so you get about 4 years of this reduced protection, and then it starts to fade away. About 10 to 20% of malaria infections result in what's known as severe disease. And the main types of severe disease are severe anemia, pulmonary edema, and what I'm going to talk about today, cerebral malaria. And again, the most vulnerable are the people that have not been exposed multiple times. So children under 5 years of age, and also migrants or travelers to the area. Several tens of thousands of travelers to these malaria endemic areas will contract malaria every year. So when you go from no exposure into a high transmission area, you're definitely vulnerable. And also pregnant women because of their suppressed immune system. The disease has 2 main stages to it, what's known as the liver stage and the blood stage. And so the parasite is carried in a mosquito vector. And so what happens is a person is bit by that mosquito and these sporozoites are released. Those traffic to the liver and go through replication within hepatocytes. And what's interesting is this is a completely asymptomatic or silent stage, so people don't experience any symptoms, they don't know that they're infected. Eventually, these hepatocytes burst and the parasite's released into the bloodstream, and you go through this process of infecting red blood cells, bursting those open, infecting new red blood cells, and the process continues. And this is the symptomatic stage where people will experience things like fever, chills, headaches, splenomegaly, low platelets, anemia, and hyperbilirubinemia, which you probably know more about than I do. But these are some of the common symptoms and findings when people come into the clinic feeling ill. And luckily, diagnosis is very straightforward, so it's still just a blood smear for parasites, which is cheap and effective, and you can see here that you can clearly see these ring structures on this example slide. Treatment is generally just anti-parasitic drugs, although there is more resistance, increased resistance happening worldwide, and also just supportive care for people as they're experiencing these symptoms. And particularly the progression for severe disease is very rapid, so starting the treatment as early as possible is really important. And I kind of just alluded to this. So during cerebral malaria, progression into the severe disease, which is characterized by convulsions and coma that can last for hours to days, and ultimately, unfortunately, many people death. This progression can happen within hours of the symptoms presenting. And it can also develop even after treatment and presumed clearance, or what appears to be clearance of the parasite in the blood due to an ongoing or somewhat delayed immune response that comes on. And there's a couple things in humans that have been noted as, sorry, you can't see that well at all, so I'll just have to describe it. It looks good on my computer, but one thing that's been noted is parasitized red blood cell sequestration. So that means that upon autopsy, they can see a lot of these parasitized red blood cells clogging up blood vessels in the brain. And so that restricts blood flow and is a big problem for the person. The other thing that's been noted is edema and swelling, and that's been shown by MRI studies. And so here, hopefully you can see this a little better. You can see there's like these, undulations of the brain here, and these are gone in the cerebral malaria patient as the brain is, swelled up and remove those folds. So the pathology associated with cerebral malaria is CD8 T cell driven. And this is kind of a busy slide, but I'll just point out a couple of key things. So through experimental models, we know that parasite-specific CD8 T cells get primed in the spleen in the early part of the infection, and then these migrate to the brain. And then here in the brain, they see antigen again that's being presented by endothelial cells in the brain vasculature. And so when they get activated again in this brain vascular compartment, they will do things like make interferon gamma, and they'll secrete granzymes, and this activity will cause breakage of the tight junctions between endothelial cells, and you'll get blood-brain barrier disruption. So that is the main path to demise in this model. And this type of activation of CD8 T cells in lymphoid organs and then trafficking to the brain has also been seen in viral infections. So there is a mouse model of this severe blood stage infection, which is what we work with. And this uses a certain strain of the Plasmodium, and these strains luckily are species specific. So this is actually a BSL-1 pathogen because it will only infect mice. And in the C57 Black 6 model, they pretty uniformly will succumb to the cerebral malaria about 6 days after infection. If you treat with anti-malarial drugs, for example, and they survive this phase, they will get severe anemia still, or if the clearance of the parasite is not complete or they survive this part of the disease, they'll later get severe anemia. So this is just an example of a survival curve here. So we can just infect the mouse with Plasmodium infected red blood cells intravenously. And then here's this time frame where you can see the survival rapidly drops off about a week after the infection. Here's if you give robust and continued anti-malarial drugs, they will survive. And other things that have been noted that are required for the development of this cerebral malaria, which here I'm calling ECM, which stands for experimental cerebral malaria. CD8 T cells are required for the development of this disease, and you can even deplete the CD8 T cells on day 4 after infection, and the mice will still survive at that point. Also, if you get rid of interferon gamma or perforin, those effector molecules that CD8 T cells are using in the brain to cause destruction, that will prevent disease. And trafficking molecules like CXCR3, that's what CD8 T cells are using to migrate to the brain. So if you get rid of that and disrupt that process, then the animals will survive. So we were interested in thinking about ways that this pathology by CD8 T cells could be modulated and exploring this infection model further. And so we started looking at the use of cytokine complexes to modulate the immune response. And it had been shown that cytokine therapies can influence the intensity and the duration of immune responses. And these complexes, which I'll describe in more detail in a couple of slides, had been used to extend the half-life of cytokines in vivo and also increase their signaling power. And it had been published that the use of these can reduce bacterial, viral, and tumor loads in animal models. And this is just an example from our group where we infected a mouse with a gram-positive bacteria, Listeria monocytogenes, and with treatment with an IL-2 or IL-15 complex, 5 days later, when we measure the bacterial load in the animal, you can see that compared to a naive animal, which has a very high bacterial load, treatment with these cytokine therapies results in a lowering of the number of bacteria left in the animal. So we wanted to use this approach to look at the development of the immune response against cerebral malaria, and we tried a number of different cytokine complexes, and I'm showing here kind of the most interesting combination, which was treatment with the IL-2 or IL-15 complex. So in this example, we're treating the animals prior to being infected with the Plasmodium, and this is just a survival graph where you see that without any treatment, the animal succumbs, as I showed you before. With IL-2, the animal also succumbs, but surprisingly, with the IL-15 treatment, a substantial number of animals survived that infection. Then we also performed experiments where we treated after the initiation of infection, because obviously that's more relevant if we think about a human therapy. Someone comes in and they're experiencing symptoms, and we found that we could rescue a significant percentage of animals if we gave the therapy after the infection had been initiated.
How durable is that phenomenon if you carry it out a week's width beyond that?
I don't think we have rigorously
That's
done
okay.
that.
Okay.
Yeah, my guess is it would not last forever.
Yeah, obviously.
But I don't know how long.
That's okay.
Yeah. Okay, and this was kind of surprising that we saw this difference between IL-2 and IL-15, such a pretty dramatic difference, because both of these cytokines and these complexes signal through CD122 and CD132, which is the IL-2-15 receptor beta and gamma chains, okay? And so there's another receptor for IL-2, which is the IL-2 receptor alpha chain, but this complex blocks interaction with that. And so both of these are going through similar signaling or similar receptors on the cell surface. And in this case, I'll just point out this one's bound to an antibody, and this one is bound to the IL-15 receptor alpha subunit. So there is a little bit of difference there. So CD8 T cells and NK cells were known to be expanded by the use of these two cytokine complexes. And so I'm showing that here. We did that again, and you can see that treatment with either one expands CD8 T cell numbers and NK cell numbers quite a bit in the spleens of these animals. And that makes sense because in particular, memory CD8 T cells express high levels of the CD122 receptor, and also NK cells as well. So they express much higher levels than, say, B cells or even CD4 T cells. And so we started thinking about NK cells and CD8 T cells and how they might have been modulated by these treatments. And in this experiment, we started by depleting NK cells from these animals. So you can see here, this is when we did the NK cell depleting treatment. So starting before the infection and then one more after the infection had initiated. And what we found was that if we depleted NK cells, the mice now succumb to the infection even when they were treated with the IL-15 therapy. So this is the positive control where we treated just intact animals and they survived. Secondly, we performed an experiment where we treated an animal and then fished out or isolated the NK cells themselves and transferred those to a new animal that was then infected. So in this way, we're isolating the effect to just the NK cells that have been treated with cytokine therapy. And here we saw that with transfer of NK cells treated with the IL-15 complex, we got that protective effect. Transfer of CD8 T cells did not do anything, as you might expect from what I've told you of their more pathogenic role in this disease. So for the first part, I'll just summarize to say that the IL-15 treatment, but not IL-2, prevented the development of this experimental cerebral malaria. And we could also do a therapeutic administration after the onset of infection. And I didn't really highlight this, but we also were able to show that IL-15 treated mice have reduced edema and reduced blood-brain barrier breakdown. And that, importantly, the IL-15 complex treated NK cells were necessary and sufficient for to prevent the development of ECM. So then we wanted to delve into this further and think about how is this happening. So these NK, yeah.
So, sorry, just a quick question. If you treat NK cells ex vivo with the IL-15 and then put it into the mice, are they protected? Or do they have to see the IL-15 in vivo?
We haven't done that. So I don't know.
You want to guess?
My guess is it needs to be in vivo, that that's going to be important. But I haven't tested that. So first we looked at just simple parasite load to see if that was reduced after this treatment, and we didn't really see any difference between percent of infected red blood cells in untreated or treated animals. So it didn't seem like we were just, minimizing the parasite load in these animals and that was preventing disease. The other thing we explored were CD8 T cells, since those are, primary drivers of pathology here. And we thought that there might be a reduction in total numbers of CD8 T cells, but this data I'll show you showed us that treatment with the IL-15 complex did not prevent the expansion of the cells. So here I'm showing some plots gated on CD8 T cells and then using a tetramer reagent that's specific for, a T cell specific for Plasmodium antigen. And then here's numbers of cells in the spleen, and you can see that if anything, there's more T cells in the spleen, and in the brain, we really didn't see any difference in total numbers of these populations. So it didn't seem like the CD8 T cells were being killed or failing to traffic to the brain or something like that. However, we did see some reduction in activation, which I'm not showing, and interferon gamma, which I'm showing here. So this is using a reporter mouse. And then we can kind of quantify that here to show that both the mean fluorescence intensity of the YFP here, which is registering the interferon gamma production, and the frequency and number of interferon gamma positive cells in the brain was somewhat reduced with this treatment, as shown here. So it seemed like there was some reduction in, not numbers, but in the activation and the production of effector molecules like interferon gamma by these CD8 T cells. So next we wanted to know, how are these NK cells mediating this change? How is this coming about? And to do that, we look to RNA sequencing analysis on cytokine-stimulated NK cells. So here we treated with either IL-2 or IL-15 in the mouse. Then we isolated the NK cells and just did bulk RNA sequencing. And probably due to the similarity between these two cytokines, there weren't very many genes. Only 40 were greater than twofold different between these two groups, which is kind of nice in a way. And so one that we highlighted from this list is IL-10. And about the time that we were working on this, a paper came out in Immunity showing that IL-10 was able to directly inhibit CD8 T cell activation. And we had kind of seen a reduced antigen sensitivity by CD8 T cells from the brain. Both, I just showed you, they were making less activated, less interferon gamma. And also if we did a peptide titration, we could see that their sensitivity was somewhat reduced in the brain after this treatment. So it turns out that NK cells have been shown to make IL-10 in a number of different contexts. And particularly during systemic infections and even after adjuvants. So in this paper, they're using a reporter mouse, which we use as well because it's pretty difficult to stain for IL-10 in the mouse. And so you can see here after a number of different infections, the NK cells are scoring positive for this GFP, which relates to the IL-10 production, and even after things like LPS and CPG. And we had also shown, using Listeria infection again, 4 days after infection, we could see using that same reporter mouse that there was a pretty high frequency of NK cells that were IL-10 positive after a virulent infection and kind of less so after an attenuated infection. So IL-10 is known to be an anti-inflammatory cytokine and a way that the immune system can follow a wave of pro-inflammation with this anti-inflammatory cytokine to kind of calm down the immune response. So then we went back to our treatments, and we saw that this is without infection here. This is just treating the animals with either IL-2 or IL-15 complex. And you can see that in all of these compartments of the animal, we get quite an upregulation of the IL-10 GFP signal. And if we look at other immune cells, we can see that CD4 T cells, we aren't really picking up any signal there. And there was a small fraction of CD8 T cells that seems like they might be making IL-10. Additionally, if we went back to the infection model and we compared untreated to IL-2 and IL-15 treated, we could see that there's, particularly by day 6 post-infection, there is a large frequency of NK cells that are making IL-10 in all of these conditions. However, when we treat with the IL-15 complex, this large frequency is there from the get-go. So I think that, the NK cells are capable of turning this on and making it normally during infection, but it's too late because by this time, by day 6, the animals are already suffering the severe consequences of the disease. And I also wanted to point out that despite this making IL-10, the cells are still capable of making interferon gamma in this situation. So and we don't really see any alteration of this ability with any of the treatments. So then if we go back to our model of ECM and think about our treatments, here we infected IL-10 knockout mice, and you can see that if you treat IL-10 knockout mice, they are not rescued from this infection. And then sort of the final best experiment we treated wild type or IL-10 deficient mice with the IL-15 complex therapy, and then adoptively transferred the NK cells from these animals into new mice and infected. So in this way, both the wild type and the knockout have been treated with the complex, but in one case, the NK cells cannot make the IL-10. And we were able to show that there was a similar response to the complex in both of these animals. So the only difference should be the production of the IL-10. And when we did this experiment, we saw the blue line here, or the IL-15 complex treated NK cells from the knockout mice. You can see that they lose the protective effect that they achieve with the wild type NK cells. So for this second part, I have shown you that after this IL-15 treatment, parasite load and CD8 T cell numbers are largely unaffected, but we did see some reduction in CD8 T cell activation and interferon gamma production. And we found that IL-15 treatment induces NK cells to make IL-10, and it's this feature that's required for the protection from the experimental cerebral disease. And that the NK cells make IL-10 just normally during the untreated course of infection, but it's not until day 6 when symptoms are becoming very severe.
So, question.
Yeah.
Does the IL-10 have to be NK cell derived, or do you have any data? There are so many other cells that produce IL-10. So do you know whether IL-10 from any source is going to give the same?
I have not done that, but you're right that IL-10 can come from other cells. And in humans, they think that IL-10 coming from CD4 T cells, that has been shown to be important and associated with like a decrease in symptoms. So it's probably the cytokine and not the particular source, although there may be unique features of the location of the NK cells and the CD8 T cells together, and that's something that we're in the same area, and that's something that we're interested in.
And one other quick question. So from comparing the differences in gene expression between the IL-2 treated and IL-15, there were 40 genes that were differentiated. So looking at those, apart from IL-10, are there other anti-inflammatory genes that were different?
We really need to go back and look at some of these other genes. CTLA4 is there.
Right. Is that granzyme?
Yeah, granzyme K here. I haven't looked at this list in a while, to be honest. But it would be worthwhile to go back through it.
It would be nice to know whether there's just more than one anti-inflammatory way that.
I'll say it didn't jump out that everything was an anti-inflammatory signature on the list, but there certainly could be other things contributing to this. Okay, so now I just wanted to say a few things about kind of our ongoing studies and the directions that we're going next. So we have this model where NK cells are making this IL-10. And one question we have is what cell type or types are responding to this NK-derived IL-10. So you can envision that it could be directly on CD8 T cells, but it could also be on the endothelium in the brain. And it's been shown that in culture, if these cells are exposed to IL-10, they'll downregulate MHC class 1, for example, and co-stimulation. So that could be a way that the activation of the T cells is getting reduced. It could also be something early that's happening in the spleen. So in general, we're interested to know what what of these many cell types could be responding to this cytokine. And the way that we're approaching this is to use Cre-specific deletion of the IL-10 receptor alpha by crossing, Cre-specific mice with flox mouse. And so in this example I'm showing here, this is a CD4 Cre, which will delete IL-10 receptor from both CD4 and CD8 T cells. And you can see that we get kind of a partial effect, where we lose some of the protection, but not all of it. So this could be sort of a complicated situation where multiple cell types are being affected. But we have breeding going on now, so the way we're it's a lot of mice, but the way we're pursuing it is to do this specific IL-10 receptor deletion on either endothelial cells, CD8 T cells, even NK cells could conceivably be responding to the IL-10 that they are themselves producing, and dendritic cells. Secondly, we'd like to know if the NK cells, it's kind of related to this question, do they need to reach the brain to exert their protection? And so one molecule we're thinking about here is CXCR3, because CD8 T cells are highly dependent on that molecule to reach the brain. So we've just done a couple of experiments where we've co-transferred wild type and CXCR3 knockout NK cells. And this is just showing cells that made it to the brain, basically. And you can see there is a fair reduction with the knockout. So the next step will be to see if that prevention will prevent the animals from surviving the infection. So that if the NK cells can't traffic to the brain, will the whole thing be disrupted, and now the mice will succumb to the cerebral disease. And then lastly for this part, we're interested in doing some imaging in the brain, which is a totally new area for me. But one thing that's been looked at in this model is the movement of CD8 T cells within the brain vasculature. And you can actually see how quickly they move and the arrest of the CD8 T cells along the endothelium. And greater arrest tends to mean that the CD8 T cells are seeing antigen and they're staying there and they're doing negative things in the brain. So, and that's just kind of here to remind you how they're seeing antigen and getting signals from these endothelial cells. And here's an example picture showing CD8 T cells along the vessels in the brain. And so there is some work out there about the movement of CD8 T cells in this model, but nothing has been done about NK cells. So we'd like to add the piece of visualizing all of these cell types, CD8, NK, and the endothelium, and looking at their interactions. Okay, lastly, I wanted to get back to thinking about humans, where I started, and talk a little bit about a human therapy, and we just have a little bit of data with human NK cells. So we also have done a few experiments using this ALT803 compound, which is a human version of the IL-15 complex. And we found, shown down here, similarly to the IL-15, this ALT803 also induced IL-10 production in the mouse. And it protected the mice not quite as well as the mouse version, but we saw some efficacy in our experimental model. And this compound is in phase 1 clinical trials and being thought of as a treatment for a number of cancers, either singly by itself or in combination with other therapies like anti-PD-1. And it's being used for its ability to increase the number of NK cells and CD8 T cells and cause their pro-inflammatory effects of cytotoxicity and interferon gamma. And then we can also get human NK cells to make IL-10. This is showing an ELISA after a lot of different combinations of the alt-803 and these cytokines. It turns out that IL-15 alone will not get human NK cells to make IL-10, and it takes a combination of this cytokine cocktail for the cells to really robustly start to produce the cytokine. And we've also been able to see this by doing assays like real-time PCR. We've seen it by single-cell RNA sequencing, but we've had a lot of difficulty measuring the IL-10 in flow cytometry, which we really have been wanting to get to. So a few weeks ago, we were able to get our sort of first positive staining using anti-IL-10 antibody. And this is just showing here, we get very low background with just the IL-15 and with the cytokine cocktail. This is after a few days in culture of purified NK cells. We can start to see some positive staining here. So we would like to use this assay and move toward this to ask questions like what types of NK cells are making the IL-10, what are their phenotypic characteristics, and ultimately then we would like to, So I wrote this down. What type of NK cells are making IL-10, and is there a correlation between NK cell IL-10 and protection from malaria? So this I'm showing you, this is just a US healthy person sample, but ultimately we'd like to look at people infected with malaria. And the way we are approaching that is we have a collaboration with Peter Crompton at the NIH. This is Peter here in Mali, Africa, where he has had a cohort for quite a long time. And in this area, they get really intense seasonal exposure. It's sort of interesting. In the rainy season, greater than 90% of individuals will carry parasite in their blood. And then in the dry season, it'll go to virtually zero. So the people go through this cyclical pattern every year. And he has followed people for many years now and has sampling then before the rainy season, during the onset of malaria symptoms, when people come into the clinic, and then after resolution. So we would like to utilize these samples to look at NK cells and their ability to produce IL-10, and then correlate that with the already collected clinical parameters that they have. So just my overall take-home points for my seminar are that mouse and human NK cells can make this anti-inflammatory cytokine of IL-10. I think we normally think of NK cells as these pro-inflammatory interferon gamma producing cytotoxic cells, but they also have this feature. And that treatment with IL-15 encourages NK cells to proliferate and to make IL-10, and that we found this NK cell-derived IL-10 is sufficient to protect mice from this severe malaria disease. And so the significance, I think, for malaria is that NK cells have both these pro and anti-inflammatory features, and really the immune response during malaria, the goal is really to decrease parasite load, but also keep that inflammatory response in check so that these overt immune responses don't get out of control. And so I think NK cells may be a unique cell that they can potentially participate in both of those features. And also, I think beyond malaria, a better understanding of these pro and anti-inflammatory features and how they're induced is really needed to optimize these therapeutic strategies. So if we're thinking about, using this to treat people in a cancer immunotherapy context, and we really just want the pro-inflammatory features, are there ways that we can kind of induce either side of the coin to our advantage? And with that, I will acknowledge the people that did the work that I showed you, and also our ongoing collaborators, both here and at the NIH, and funding sources. And with that, I can take more questions. Thank you.
So what about the people who have sort of these chronic exposures and they do not get cerebral malaria? Is that because they have a reduced parasitic load, or is that because of modulation of the immune system?
I think both. It's kind of hand in hand. I mean, they definitely will, over the years, have less parasites, but it's not like a real one-to-one where always a person with a high load will get severe disease and a person with, a low load won't. So I think it's a combination of factors there. And definitely they have a lower, less cytokines in the serum, lower inflammatory indicators with age.
I'd like to add to Dr. Clark's point. In the laboratory, we diagnose malaria not infrequently, and to my surprise, high parasitemia does not. I always felt that was the biggest driver of developing cerebral malaria. So about 2 years ago, Minnesota man goes to Tanzania, never been to Africa before, gets misdiagnosed, comes back almost in coma, extraordinarily high parasitemia level. He dies of cerebral malaria. Recent native of Togo goes back there and gets infected, comes back and has similar high parasitemia, does not develop cerebral malaria. So I've had to rethink about all the contributors that can modify high parasitemia and prevent the development or alter development of cerebral malaria. And the only thing I can come up with, other than the points you make of inflammatory molecules, is the presence of antibody from previous exposure that may modify and mediate some of the protection. And so, in your elegant experiments, I wonder if the addition of any specific antibody might glean anything interesting. And then secondly, can you further try to translate your findings to applications to humans who are at risk of cerebral malaria before it develops even?
Yeah, I mean, that's interesting, the case that you mentioned, and I would say that we've seen the same thing in the mouse, where, sometimes you'll have one with really high parasitemia, and it doesn't always mean severe disease, and vice versa. Antibodies definitely important, and NK cells can mediate antibody-dependent cellular cytotoxicity of infected red blood cells. That's one way that they can work, but there's a lot of ways that antibodies can help to, remove parasites from the person. So, yeah, I think you're right. That's definitely a critical part of the protection in people. Absolutely.
Sara, as a sort of a follow-up of Pat's comments and along the line of some of your ongoing experiments, I want to start by asking you a question. Have you seen the same plugging of capillaries in other parts of the body similar to the plugging of capillaries that you showed us in that one slide? And the reason I'm asking this is that I'm wondering if there's something unique about the endothelial cell in the brain that allows this to occur primarily in the brain, because we know there's a lot of plasticity to endothelial cells, and obviously the endothelial cells are part of the blood-brain barrier, which makes it unique to other parts of the body. Do you have any comments on that?
I have not looked at that. Other people have, and it has been commented that that sequestration piece is not as prevalent in mouse as they think it is in humans, as has been seen with the autopsy stuff in humans. So that could be a feature of the different Plasmodium strains and the different models. I'll also say that the other place that you get like this breakdown problem is in the lung. So it could be that, like you're saying, features of the lung and the brain are particularly vulnerable to, attack during this process, and maybe there are parasite-infected red blood cells that get more hung up in those areas and kind of, start this cascade. Yeah, Harry.
So back to your dirty mice. Could the gut microbiome be impacting the inflammatory response?
Sure. It impacts everything, right?
I mean, could you do the same experiment in a dirty versus a quote clean mouse?
Yeah, I should have included that. I have done that. I haven't done the IL-15 complex treatment. I've just done infecting a dirty mouse. And most of them still get cerebral malaria. There is a little bit of an increased survival over just a SPF housed B6 mouse, but most of them still get disease. But I am interested in thinking about that.
You need some mice from
What?
You need some mice from Molly.
Yeah, I'm interested in following up on that.
But in your Molly experiments, if you're working with any of those humans with malaria, you absolutely must have serum so that someone can measure antibodies as well.
Yep. Yes, absolutely.
So the situation in malaria in the brain is reminiscent of cytokine release syndrome in human patients treated with T cells, cytotoxic T cells.
Yeah.
So in patients, what is used is antibody blocking interleukin 6, which sort of, does that work in the malaria?
I don't know.
And there's much study about the role of macrophages in the CRS, so I wonder if there's been a role for macrophages in this situation here.
Yeah, that's interesting that you bring that up. I was just reading one of Chris Pinnell's grants about that exact release syndrome in the brain, and you're right, it really made me think of this model as well. So I should think about that more. But
And also when you show those clogs, I mean, do you see those clogs in the animal system?
I haven't looked for that specifically. I just know that other people have commented that they are not as apparent as they are in humans.
Yeah,
But
I wonder if there
myself,
are macrophages in those clogs eating those red cells.
yeah.
It looked a bit like that. Yeah, just one small picture.
I think there's definitely a role for antibody protection in those with chronic malaria.
Yeah,
I think it's
absolutely.
I'm not sure whether the mechanism is pro-interleukin 6 or something else, because you could always predict who's going to get cerebral malaria. It's almost always the people with the very first infection, that's babies or experts who have never been to malaria area. And like Pat said, sometimes you see, and I used to look at blood smear when I was in Nigeria a lot, and it's amazing how much parasitemia you could see in patients with hemoglobin of just 5 grams, very low, very anemic pale, but they never get cerebral malaria. They might get big spleen, sometimes rupture, and that's rarely cerebral malaria. So I think there's a role for preformed antibodies protecting against the cerebral. Now, the exact mechanism, I'm not sure whether it's got to do with interleukin 6 or not.
Yep. Okay. Thanks, Sara.